I'm Mark Geyer from NIH. This is a session on national initiatives, and I will just start by telling you what the speakers were asked to address in a short 5 to 7 minutes. And they were asked to talk about their national initiatives, addressing questions: how are you leveraging or planning to leverage the Global Alliance in your national initiatives? How do you plan to share data or support data sharing? And have you identified major barriers that the Global Alliance is currently not trying to address? And or are there challenges that are unique to your jurisdiction that the Global Alliance should be aware of? So those were the questions that people were asked to address, and we have 6 presentations, which we'll try to get through efficiently. We will not take questions after each talk, but then at the end, the panel will come up, and we have been scheduled for about 15 minutes for a question. So with that, let's get started. The first speaker is Maurice Swertz, talking about the genome of the Netherlands.
Thank you very much. So I'm happy to be here and to tell you a little bit about the Genome of the Netherlands, which is a project of the Dutch BBMRI, the Dutch Biobanking Consortium. I was supposed to be in 5 minutes, so I have 5 mini presentations to bring your attention to a few relevant genomics activities I think you can work with and where we can synergize. And I will run through them quickly as promised. So first is BBMRI-NL. I don't know whether all the people from abroad are aware that there is a European biobank infrastructure called BBMRI. And that is a hub and spoke infrastructure similar to Elixir, where each national node can set up a national infrastructure and then federate that into Europe. So the BBMRI-NL is the Dutch one, and our remit is to harmonize and enrich biobanks such that you can integrate the data and provide easy access to all this data. And some numbers: there's now 900,000 samples in the Dutch biobanks, which is about 5% of the total population. And about 20% of those are population biobanks, so general population, like deep, large registries like Lifelines, where there's like 160,000 individuals captured. And then there's many clinical biobanks, which accumulate to 80% of the total number. And they may be very shallow or they may be very deep. That really depends on the biobanks. And there's about 200 of them. And when we are not counting the small ones, so the rare disease, we didn't count it to this 200. So we got 30 million from the Dutch government to work on this in a period of almost 10 years. And the first phase is almost finished. And there we decided to do a lot of broad projects, which we called Rainbow Project, or abbreviated RP. And in the second phase, we want to consolidate this into one infrastructure. So the first was more like this, second phase is like the harvesting phase. So I only will talk about a few of them that are relevant for genomics. And the first one of them is the Genome of the Netherlands project. So as far as we know, it's the first population-specific, like a country, that did a whole genome sequencing, and we published it last year. And this is how it's made up. So 4 large population biobanks contributed samples, which accumulated to 769 individuals. Originally, we wanted to have 750, but then we thought it would be nice to include some twins, which upped this number to 769. And we sent those to BGI to have a 12x coverage sequencing, which at that time seemed to be the best value for money. And what is otherwise special about this is that we did it in trios, which allows us to do particular types of research, but it's also very nice for quality control. So what came out of it? I don't have time to really go into the research details, unfortunately. So just some numbers. So of course we found a lot of new variants, which may or may not be specific to the Dutch population, because of course we also sequenced a lot of the common ones. So that's depicted on the left. And we also did a search for more larger variations, structural variations, which are depicted on the right. So the x-axis is the variant size, and the y-axis is the count of them. And you see that we actually could discover a lot of new variants. So in terms of raw data, that's about 30 terabytes of FASTQ files that we stored in 2 locations, and also the analyzed data, which is about 60 terabytes of BAMs. in 2 locations in the Netherlands, so the 2 largest compute centers, so that there's always a place where you can access its data, even if one of them is in maintenance. So you can read all about this in the Nature Genetics paper. It actually is immediately being used for the clinic because it helps the clinicians to get rid of the common variants. And it's also often used for imputation. And then we have sort of 50 research requests since our publication where people requested data access. So I shouldn't forget to mention that there was another major impact of this, and that was that we actually united the human infrastructure in the Netherlands to work with this data, as well as the IT infrastructure to deal with all this. So that was a great benefit for us. So how to get access to it? So some of the data is public, which are the aggregated SNPs and indels and SVs, but for privacy reasons, there's a deal between the donor of the data and the biobank. We have to put the individual level access behind a controlled access. So the data is in EGA, and you can request it via an online form on this http://genome.nl. So in the follow-up of this, we do many more studies, and I cannot tell all about them, but this is one of them, the integrative study. So what we wanted to do here is add additional layers of data, which includes a lot of RNA sequencing, which in turn results in a lot of more genome information, because you can, of course, derive genotypes from them, and then integrate that with other omics data. So unfortunately, no time to talk about that, but you can read in many papers about it yet, and also on the BBRINL website. And then the third and last project I wanted to highlight is that we, of course, also collect more and more next-generation sequencing data in the clinic. So in the Netherlands, there's 8 diagnostic labs associated to the large academic hospitals that collect all this data. So there's thousands of individuals currently being sequenced, usually at gene panels, but more and more clinical exomes, and some of the time already whole genomes. So this is a thing that will become more and more important. So looking forward, we did a pilot because one of the things you cannot do currently easily is ask the question, has this variant been seen in another diagnostic lab? And of course you want to do that. And if you have seen that, is there somebody who has looked at it? So we started a project to do data sharing and answer these 2 questions. And the architecture is very simple. You want to capture the raw genotypes as they come out of the sequencing device and count them. So you can ask frequentist questions, and then at the end also look at the interpret variants, which we call the managed variant list in the Netherlands. So just to make sure that people understand what we're up to, we are counting the genotypes, and we are not counting them simply. We want to know how many homozygous or heterozygous alternatives or refs we found. So this is actually a VCF file for the people who want to know. So you can look at it in different ways. So we did a national browser using a simple package that we built ourselves. But we're now more and more looking at how to disseminate this to LOVDs and these kind of things. And one of the things we looked at is the Cavivariome, which came out of Leicester. We uploaded one of these patient cohorts for the cardio. And here you can fairly easy in an open access way find out whether a patient is actually available in this set and how often it has been seen, which means actually these numbers are important during diagnostics. You don't even need to ask access unless it's a very low number. And then finally, the diagnostic labs, they use a software package called Cartagenia to do their in-house analysis in most of the labs. So also inside of their diagnostic process, they can now see it. So each row is actually a patient that is seen in another lab, and you can compare your evaluation. So looking forward, for us, this was like the first phase, and now we need to consolidate. So we made a list of topics that we think we should work on in the Netherlands, mainly around IT. There's many other topics, but I thought here I should limit to IT. So one of the first things is to improve the access to all these samples and data. So we made catalogs. They need to become better. You need to be an easier way to request the data. You need to have all kinds of ways to interrogate the data, because in many cases we still need to keep the access private. And then of course you need like a safe haven where large multi-center data sets can become together. So that's a large challenge because that will not scale up to the whole world. So I hope that together we can come up with another architecture. And that's building on existing projects in the Netherlands. And then a mirror to that is actually the BBMRI Europe is also going into this direction, but then trying to coordinate this work across the European countries. So there's similar things there, like a directory of biobanks or where can I find samples, but also to coordinate the services that are out there for either the national nodes or the biobanks. Another thing I wanted to mention is also an LC common service that people want to coordinate with. And of course, I see many places where we could synergize, because I think there's many places where we should actually talk to each other about these APIs and share the work on implementing them if that's needed, or simply connecting to the ones that are emerging. So with that, I want to wrap up. Thank you.